Good afternoon. I want to sort of go through this ribonucleotide reductase, which at a glance seems like one of the more complicated things that's going on in this PowerPoint, but actually it's not too bad. So I've developed a model for looking at it, and I thought I'd share it, and maybe it's a benefit to you guys, and maybe I'll learn something by trying to teach this. So basically, you have two sites. This is the um, activity regulation, and this is analogous to like the on and off switch of a TV. So this just determines whether or not the whole thing is on. It doesn't really dictate what it's doing, just is it on. And then we have these, which are like the modes. So these are the different modes. Each M is a mode. And each mode would be like, your TV can be taking from the cable box or from the DVD player or from the, game console or whatever, from your laptop, whatever input you have, you can switch it and it'll do something different. But again, that's separate from the on and off control. That's sort of what we have going on here. Now, this process, of course, it is very, very highly regulated, naturally. And let me see if did he include? He did not. Well, okay, then we'll look at it this way, is that once you have processed something into the deoxygenated form, it's committed to basically DNA synthesis. It's here it is. Reduction at the 2' position commits nucleotides to DNA synthesis. And that's important because we know that ATP is very critical for the function of the cell. So going back to our model here, in order to turn this thing on, we have to have ATP, and we also have to have enough ATP that we don't need to use this for something else. So we know that this is under cell cycle control, and you have the growth phases, and you have mitosis and all that, and there is S phase, and S phase is the synthesis phase of regulation. I think he lists those here. Here they are. So you have growth phase 1, S phase, G2, and M. So the cell has these checkpoints, which basically analyzes, well, what's going on? Do we have enough energy to be growing and synthesizing DNA, or do we need to sort of double down and try to stay alive because we don't have much energy? So if the cell is entertaining the idea of going into the synthesis of DNA phase to prepare for mitosis and splitting and creating more cells, there's going to be a check, and that check is going to be, do we have enough ATP, among other things, but one of the things is, do we have enough energy? And the actual mechanism by which that check occurs, there may be others, but one of them, it occurs to me, is that we need enough of a concentration of ATP to turn on the enzyme. And it's logical. Same thing, if you're living on your own and you need money. So if you have a lot of money, then you might think about remodeling your deck or upgrading some facet of your car or something. But if you don't have enough, you're going to commit that to feeding yourself and buying clothes and school supplies and such. And that's the same thing here. So if we have enough ATP to spend on renovation and upgrading and dividing, we'll do that basically. Okay. And then there's the other part of this where you can have, you turn it off when we have DADP. Either DADP or is it ATP? Okay, it's DATP, sorry. So DATP. And the reason for that is because basically when we go through this whole process, one of the last things that we're going to synthesize, according to his PowerPoint, is DATP. So when the DATP concentration is rising, it basically means that everything is sort of in balance, we have enough of everything, we're good. Let's turn the machine off, let's save power, let's save ATP, let's conserve on the electricity bill. And so the whole enzyme is turned off by DATP. And then there's the other part of this where you have the mode basically, which is, you know, what are we going to allow? So this would be here. Let me label this. This is the active site. Active site. And you can imagine sort of it being a machine and just going through. In reality, it comes in, it comes out. We know that, but let's say this is the active site, and basically the different modes will each represent a different product or a different substrate that can come in here. So here, you know, we might get, if we look at the list, you know, we might have, so that would be the catalytic site, the active site. And we might have GDP going in, ADP, or CDP, or UDP, which are the ribonucleotide form. And if they're going in, then we can convert them to the deoxy ribose form. And that's what happened here. So we have step one, ATP occupies activity site A, so the enzyme is turned on. We spoke about that. And then ATP can also enter the specificity site. So what that means is that ATP it'll be here, but it'll also be over here. So if we have ATP here, then this will activate the mode wherein we synthesize or we accept CDP and what was it? Was it GDP? Was the other one? Sorry, UDP. UDP. So uridine, uridine diphosphate or cytidine diphosphate. And so those will come in here, and what we'll get out on the other side, and if we just underline them sort of as we're making them, we get DCDP and we get DUDP. Now DUDP we've discussed in class, you know, for various reasons is just not a good idea to be using uracil and making DNA. So DUDP turns into DUMP, and that turns into DTTP, which is important because DTTP will activate the next mode, basically. And let's see what the next mode does. So we have, again, so the CDP is good, UDP is good, and we will create TTP from it, DTTP. Occupies the specificity site, and it'll favor, oh, and I should be specific, this whole thing is the specificity site. This whole shebang over here, this is the specificity site. Okay, so there are not four of them, there's just one specificity site, as I understand it. So at any rate, we get DTTP in there, and then it favors GDP or ADP in the catalytic site. So going back here, so then we get GDP and ADP in the catalytic site. So now we're going to get these two output. So okay, awesome, that's great. Now we have basically everything we need. We have UDP, which will be converted into this, but we have this. So basically, the job of this enzyme is done. You know, we're good to go. Do other things. We need energy for other things. We can turn this thing off. So, right. So what's going to be made first, apparently, is going to be the DGTP. So again, the DGTP come in here once again, and that would be the last. That would be this. That would be to DGTP, and that could come over here and do the next mode, right? And you can have however specifically this goes on. I hope the analogy is sort of sticking. And so DGTP will be in site S, and that will favor ADP entering the catalytic site. So so again, we could have ADP being made here, but this is going to mean just make ADP basically, and then we create that. And there are kinases, there are enzymes in in the environment which will convert the DPs to TPs, so we'll get DATP, which will replace ATP in the activity site. So finally, ATP will leave, and then the ATP will take the DATP will take over from the ATP, and it'll turn everything off. And so I think that's sort of the gist of what's going on here, and it makes sense. By doing that, we make something until we have we make something until we have enough of a concentration of the product that product can come and affect the specificity. Then we make something else, and then we make something else, and then once we've made everything in the right amounts that we need, then the final product turns off the entire apparatus, and we're done, and we're good to go. Let's see if there's anything else. I guess I'll cover this now. If you remember, I think it was, I forgot what it was called, pyruvate decarboxylase, I believe, from Biochem 1. We had a similar thing where we had to have this disulfide bridge mechanism going on in the enzyme. And so we had to regenerate it every time. And that's exactly what we have to do here as well. So you can see that in order to go from NDP to DNDP, this is a reduction reaction, so we need this right, so the electrons have to feed across basically, and we feed the electrons onto the DNDP so that we can get rid of the OH and attach a hydrogen. So the electrons come from NADPH as usual, and these go to FAD, which goes to FADH2. Those electrons then go here, so there's 2 electrons here, and that goes to break the disulfide bridge, and we get SH, and then that goes from thioredoxin reductase to thioredoxin reductase to thioredoxin. And so now again, so the electrons have gone from here to here to here to here. Now the electrons are here, and they go to ribonucleotide reductase. So now we have electrons here, and then we feed the electrons onto the NDP by some organic chemistry type mechanism, and we get DNDP. And of course, by going through this process, the electrons are passed, but also as they're passed, each of the intermediate steps are regenerated here. And so everything is the way it needs to be to accept electrons and sort of keep passing it on, in the same way that the pyruvate decarboxylase or dehydrogenase, I can't remember which one it is, that thing worked in Biochem 1, so it's a similar thing here. I hope that was sort of helpful. I've been trying to have a coherent way to wrap my mind around this, so I thought I'd share it with you. Yeah, so I think I'll leave this off here, and if you have any questions or thoughts, please let me know.